Okay, welcome back. And let's move to the next session. And in the next session, we will have our moderator, who is Professor Shida Xu. Is it Professor Xu, please?
Yes, this distinguished guest, dear colleague, ladies and gentlemen, the Association of Microbiomic and Metabolomic in gut-lung axis and childhood asthma had got great advances in recent years. Today's lecture is chaired by me, Sida Xu, Professor of Pediatrics, Allergy, Immunology, and Pulmonology, Mackay Children's Hospital. We will have about 20 minutes for today's talk. It is my great honor and privilege to introduce you the speaker of today, Professor Zhiyong Qiu. Professor Zhiyong Qiu came from Chang'an Memorial Hospital at Linkou. He is the professor of pediatrics pulmonology. He had finished his PhD training at the Imperial College London. He is a co-principal investigator of a first cohort study in the prediction of allergy in Taiwanese children, H. Jones study initiated since 2007 to investigate the epidemiology and the predictor vector of asthma and allergy in Taiwanese children. His topic of today's lecture is an integrative analysis of microbiomic and metabolomic for childhood asthma. Let's give a warm welcome to Professor Zhiyong Qiu. Professor Qiu, please.
Hello, everyone. I'm Zhiyong Qiu from Chang'an Memorial Hospital, Taiwan. It's really my honor to be here to share my work. The topic that I want to talk about is an integrative analysis of microbiomics and metabolomics for childhood asthma. There's no any conflicts of interest disclosures for my talk. We know about the Human Microbiome Project was launched in 2007. For being a pediatrician for childhood asthma, I really want to talk about the airway microbiome and their association with the asthma. So the first question is, what do we know about the airway microbiome? This slide shows the development of nasopharyngeal microbiome since birth to the age of 1. Then we can see the airway microbiome changed according to the age and getting to be more stable at the age of 1. We know there were a lot of host and environmental factors that could change the respiratory microbiome, as shown on this slide. For example, the feeding type, siblings, daycare centers, vaccination, infection, or smoking as well. A lot of allergens has also been reported to be associated with rhinitis and asthma, especially the house dust mite. They are quite important for the asthma and rhinitis, especially in children. So the first question is, what's the relationship between the airway microbiome and allergies? As we described before, airway microbiome maturation process appears to be more stable during infancy. However, a higher prevalence of asthma develops after age 2, and the asthma is associated with the allergen sensitization, especially for the house dust mite. So we hypothesized that the dysbiosis of the airway microbiome could potentially influence or the progress to the asthma. And we try to evaluate the relationships among the airway microbiome and the TOPI index, especially for the IgE levels and the childhood asthma. So we designed a case control study and use the Illumina HiSeq platform to identify the airway microbiome. Then we collect the children with asthma and rhinitis and health control around 3 to 5 years old and collect their short to identify the DNA from the samples and using the 16S RNA amplification to identify the airway microbiome. This is the microbiome analysis flowchart. We use the TRINE software to identify the OTU levels of airway microbiome and further analyze the diversity of alpha and beta diversity, and finally identify the different abundance between the healthy control and asthma. This is the result of the epidemiology characteristic of children that enrolled in our study. There were around 30 children with asthma or rhinitis or healthy control. As we can show on this slide, not very surprised that the children with asthma and rhinitis had a higher prevalence of mild sensation or food sensation, or either the IgE level as well. This figure shows the richness and diversity for children with allergen sensitization for mild, for food, or IgE levels. They found that there was a significant lower richness and diversity in children with mild sensitization, but not for food or IgE levels. So we can see there was a strong association between the airway microbiome and the mild sensitization, but not for food sensitization. We further analyzed the richness and diversity in children with mild sensitized rhinitis or asthma. As shown on this figure, in children with mild sensitized rhinitis or asthma, they had a significant lower richness and diversity of airway microbiome in comparison with the healthy control without any mild sensitization. So there were maybe a modulation between the airway microbiome in response to the mild sensitization, and this modulation may contribute to the development of rhinitis or asthma. The result that we studied before, we just published in a scientific report in 2017. The conclusion is that airway microbiome diversity is inversely associated with the mild sensitization, rhinitis, and asthma, especially in the early childhood. As we mentioned before, that there were a lot of host or environmental factors that will contribute to the development of asthma. For example, the host diet, or the airway microbiome, or the house dust mite sensitization. So we just want to know the metabolome associated with these factors and their relationship to the development of asthma in preschool children. So the next question is that we just want to use the metabolomics analysis to find some biomarkers for the childhood asthma. This is the study population and study design for our study. We collect the urine samples because the urine were not very invasive and easy to collect, especially in children. And we try to analyze the urine metabolites by using a time series urine sample from the birth cohort. So we collect around 30 children with asthma and 30 children with healthy controls, and collect their urine sample from age 1 to age 4. And during this follow-up period, some people will have asthma development and some people just still be healthy. So this is the flow chart for the metabolomics analysis in our study. We collect, firstly we collect the urine sample and use the NMR analysis, and we identify the metabolites in the urine, and then we classify all the metabolites into the children with healthy and the asthma. And then we try to use or identify the different metabolites between the asthma and the healthy control, and collect all the metabolites to the metabolic pathways and try to analyze and correlate to the clinical condition as well. So this is the result of metabolites that we found could be interacted with age and the asthma development. And there is not very surprised that a lot of metabolites were changed by the age because the children have different diet at different ages. But mostly important, there were 4 metabolites that may be quite very strongly associated with the asthma development. For example, the dimethylamide and the 1-methylnicotinamide and the anatonin and the guanidinoacetic acid. We further analyzed the relationships between these 4 asthma-associated metabolites and the allergen sensitization. Then we found the guanidinoacetic acid is quite common for all allergen sensitization. However, the dimethylamide was quite unique associated with the asthma, but the 1-methylnicotinamide and the anatonin were also strongly associated with the mild sensitization as well. So we know there was a strong association between the urine metabolites and the mild sensitization, and this could be contribute to or influence the asthma development as well. This figure shows the metabolic pathway of 4 metabolites associated with the asthma. And we found the 1-methylnicotinamide were categorized from the nicotinamide in the gut, and the anatonin were from purine, and the dimethylamide and the guanidinoacetic acid were from the choline metabolism in the gut. The most important, we found the gut microbiome was strongly associated with the anatonin and the dimethylamide development in the gut. So we think the gut microbiome may play a very important role to development of asthma, especially in children. We just published our finding in 2018. We found that longitudinal urinary metabolomic analysis, we can find some metabolites were strongly associated with the asthma development, especially in early childhood. So the next question, we just want to move to what's the relationship between the gut microbiome and the allergies. So again, we designed a case control study and use the Illumina HiSeq platform to identify the gut microbiome and their association with rhinitis and asthma. So we collect the stool sample from the children with asthma and rhinitis and health control. And we use the 16S RNA amplification and try to analyze or identify the gut airway microbiome that were different expressed between the asthma and healthy control. This is the result of gut bacterial richness and diversities among children with atopic disease. Although there were relatively lower diversity and richness in children with rhinitis and asthma, but there were no any significant difference among the atopic disease and the allergen sensitization. So we found that there were no any association between the gut microbiome and the mild sensitization, and no any association with the food sensitization as well. However, we still try to identify the differences of gut microbiome between the asthma and the healthy control. And we found the Firmicutes and Protea bacteria in final levels were quite a difference between the asthma and healthy control. And we also found the Escherichia and Clostridium and the Eubacteria in genus levels were also strongly different in children with asthma and health control as well. In this study, we also analyzed the IgE level in the stool, and we found a very interesting finding that the IgE level in stool were strongly associated with the mild IgE levels in the blood and IgE level total in the blood as well. So next we try to correlate these IgE levels and the gut microbiome that we identify associated with the rhinitis and asthma. And we found the Clostridium, Eubacteria, and Escherichia, these three gut microbiome associated with the asthma were strongly associated with the mild sensation or total IgE levels or the IgE level in the stool as well. So we conclude that the gut microbiome dysbiosis is strongly associated with the allergen species IgE levels, especially in children with the rhinitis and asthma. Then we just published our finding in 2019. Because we found some gut microbiome were strongly associated with asthma, how about the metabolites in the gut? So next, I tried to link the gut microbiome and the gut metabolites and investigate their relationship and the contribution to the asthma. So we identified a lot of gut metabolites and tried to classify all these gut metabolites into 6 classifications by using their functional metabolic pathways. For example, a lot of gut amino acids were classified to the amino acid metabolism, and some metabolites, for example, the beta-alanine or hydroxybutyrate or butyrate, were classified to the metabolic pathway associated with the microbiome. For example, the beta-alanine metabolism and the butyrate metabolism. And we also classify some metabolites to the glycolysis or methyl metabolism as well. Then we further analyzed the association between the gut microbiome and the gut metabolites. And we found a lot of metabolites associated with the amino acid metabolism in the gut were strongly negatively associated with the gut microbiome. But some metabolites associated with the beta-alanine and the butyrate metabolisms shows some negatively or positively association with the gut microbiome. And we found a lot of association belongs to the Firmicutes bacteria in the Finland level. And this is a very interesting finding because we found there were a strong association between the gut microbiome and the gut metabolites, and it's quite interesting finding. We further identified gut metabolites which were significantly differentially expressed between the children with rhinitis, asthma, and healthy control. Then we found only one metabolite, histidine, was significantly different between the rhinitis and the healthy control. However, there were 5 metabolites appears to be significantly different between the asthma and healthy control, including the butyrate, beta-alanine, trimethylamine, hydroxybutyrate, and fumarate. Among these 5 metabolites, the butyrate appears to be a very special metabolite. We know it's one kind of short-chain fatty acid, and it's also a microbial-derived metabolite. And in recent days, many studies found the butyrate were strongly associated with a lot of disease, including atopic disease as well. This heat map shows the correlation between the gut metabolite and the microbiome, which were strongly associated with the asthma, and their correlation with the IgE level in stool and in the blood as well. As I mentioned before, the butyrate, which was significant associated with the asthma, and we also found a strongly correlation with the MIT special IgE level as well. Also, we found some gut microbiome that we mentioned before, which were strongly associated with asthma. For example, the Clostridium and Escherichia. And we found there was also a strong association with the beta-alanine in the gut metabolite as well. And this finding shows there was a strong linkage between the gut microbiome and the metabolites. And this modulation or this correlation may play a role for the asthma development in early childhood. We just published our finding in 2019. Then we concluded that the gut microbial-derived butyrate appears to be inversely associated with the IgE levels in response to the mild allergens. And this association may contribute to the asthma development, especially in early childhood. Well, this is the summary of our study. Firstly, we investigated the airway microbiome, and we found a strong association between the airway microbiome and mild sensitization. And this association may contribute to the asthma, especially in early childhood. Then we try to understand the association between the urinary metabolites and allergen sensitization. Then we found some metabolites in urine were strongly associated with the asthma. And we also found that these metabolites has been reported to be associated with the gut microbiome. So we further analyzed the association between the gut microbiome and the allergen sensitization. However, we didn't find any association between the gut microbiome and mild sensitization. Then we tried to have a link between the gut microbiome and the metabolite in stools. And luckily, we found a metabolite butyrate is one kind of short-chain fatty acid. It's also reported to be associated with bacteria metabolism. We found the butyrate was strongly associated with the asthma and the mild sensitization as well. This is a very, very interesting finding. Then we also find some gut microbiomes, especially the Clostridium or Escherichia, which has reported to be associated with asthma in our previous study. And we also found this bacteria have a strong association with the metabolite in stools, for example, the beta-alanine. So this finding shows that the gut microbiome also plays an important role for the asthma development, especially in early childhood. So in our study, we concluded that not only the airway microbiome, but also the gut microbiome play roles for the childhood asthma. Thank you so much for your listening. And this is the end of my talk, and I hope all of you enjoy my talk. And it will be my pleasure to answer all your questions if you got any questions. Thank you again for your listening. Thank you.
We are very enjoying Professor Chu's speech today. Let's open for discussion now. Can I ask one question for Professor Chu?
Sure.
Did you check acetate and propionate in addition to butyrate? Yes. The first question is, did you check acetate or
Yes.
propionate in addition to butyrate?
Thank you for this question. This is a very nice question because we know there are three important short-chain fatty acids, including the propionate, acetate, and the butyrate, which will have been reported to be associated with the gut microbial-derived metabolites. But in our study, we try to identify the metabolites in the guts in the store, but we didn't find another metabolite like the acetic acid or populate acid, but we only found the butyrate. So we can't evaluate the importance of the other short-chain fatty acid metabolites and their relationship to the asthma. So we just identify the butyrate, and this butyrate is quite important to the asthma development based on our study. Anyway, thank you. Hope I have answered your question.
Now, in recent update in the allergy and the microbiome and the metabolomic, we know different allergic disease associated with different microbiome and the metabolomic. And even in different allergy or sensitization, like peanut, milk, others, different, they also associate with different microbiome and microbiomic. Also different allergic sensitization or allergy. And I have one question is, can you explain more about the gut-lung axis? Its influenced by environment factor and it's associated with disease like asthma.
Oh yeah.
How they influence?
Yes, this is a very nice question actually, because we are still struggling on this project. But actually, I have evaluated the association of the gut and the lung microbiome. Actually, they don't have a very exciting correlation between the airway and the gut microbiome, because clinically they are quite different organ for the lung and the gut. So I'm not very surprised about it. I was still struggling on this lung and gut axis for asthma development, but we didn't get any result about it. So as you said that they are still very interested field and a very important field that we still don't know about it. So we are still try to understand or investigate many field about it. So I sorry about it. I can't answer all your questions right now, but I hope I can answer your question in the coming future. Thank you.
Is there any question?
Okay, thank you.